Welcome back to the flagship studio here at the 2024 JP Morgan Health Conference. I'm delighted to be joined today by Michelle Werner, CEO of Alterna, talking about the company and its innovations. Michelle, I was just reading that 300 million people around the world have a genetic disorder-based disease, and that fully 10% of those 30 million have stop codon disease. Can you explain stop codon disease and how it relates to your work at Alterna?
Yeah, that's exactly right. So 30 million people have stop codon disease. And what we mean by that is that it's any genetic mutation that stems from a stop codon, or otherwise known as a nonsense mutation or a premature stop. That's when there's an error in the code in a protein, and instead of coding for an amino acid that makes up the proteins, it actually codes for a stop instead. And what happens then is that the protein is either absent or truncated, and that is the cause of disease. We came up with this terminology, stop codon disease, in conjunction with our clinical advisors, because with the sort of dawn of a new modality, such as tRNA, transfer RNA, it actually requires a different way of really thinking about or classifying patients, because we have this unique ability to treat patients with a single engineered tRNA that have common mutations across many, many, many different diseases. The classical definitions of disease don't make sense in that case. We really have to think about patients by those mutations instead. And that's our approach that we're taking at Alterna.
You've described stop codon disease. Talk to us about tRNA specifically and how it addresses that stop.
Sure. So the tRNA is a fundamental component of the protein translation process, where its precise role is to read the instructions on the mRNA for each of the individual amino acids that make up a protein, and then identifies that corresponding amino acid in the cytoplasm and then transfers it to the growing polypeptide chain. So it plays this critical role. Without tRNAs, there just would never be a protein.
The T standing for?
Transfer.
Got
right?
it.
So that's what it is, because it transfers that corresponding amino acid to the growing protein. And what we're doing at Alterna is engineering tRNAs. And the reason why this is an important new approach is because in nature, there are no tRNAs that know how to read these stop codons. And that's why that translation process terminates too early. But with an engineered tRNA, these are precisely designed to bind to these premature termination codons. And so that translation process is restored, and the corresponding amino acid that should have been coded for instead of that error gets transferred instead. And the result is that full-length functional protein, which is precisely what patients with stop codon disease need to address their condition.
And I know patient advocacy is a personal passion of yours, and you have spent a career at pharma powerhouses developing drugs, and you're looking ahead to the clinic at Alterna. Tell us about how your approach to the clinic will take place and what your aspirations are there.
Yeah, so with this unique ability of our engineered tRNAs being able to address patients across a number of different diseases, we have this real amazing opportunity to fundamentally think differently about genetic conditions. So right now, in a very conventional way, even with novel modalities such as gene therapy and gene editing, each one of those has to take a disease-by-disease approach because they are precisely for that specific disease. Because engineered tRNAs can go across different diseases, we can address many more diseases than historically have been given attention to by the industry. And that's one of the things that I think is so powerful about this technology, because instead of having a handful, let's call it maybe a dozen different genetic diseases getting the attention of companies, pharma and biotech companies, we actually can look at 100 or thousands of different diseases simultaneously. And think about that from a patient perspective, right? If you're a patient with one of these rare or ultra rare conditions that might be overlooked by conventional drug developers, now maybe engineered tRNAs can become an option for you, whereby there might not have historically been anything available. And I think that's super important and very powerful.
Given the number of diseases that are impacted by stop codon disease, how does, when you think about the clinic, how does that impact recruiting for clinical trials?
Yeah. So it's also a great opportunity because we're taking a play from what is the oncology playbook called basket trials. And when you have a basket trial, the way that those are designed is that you have patients across a number of different diseases, but they are all unified because they have the exact same mutation. You give them the same therapeutic in that clinical trial, and then you measure the same or a similar outcome across those different patients, despite the fact that they have a different disease. Now, several oncology agents have been approved using basket trials, and we think that engineered tRNAs are uniquely suited to this approach. Many different diseases, all having the same mutation. We give them the same engineered tRNA. We measure a same or similar outcome across the board. We think this is a great opportunity because, number one, it would allow for many different types of patients to be able to have the opportunity to benefit from a novel innovation, but also have massive indication expansion potential. Right? Historically, with a traditional design, you've got one clinical trial, one indication, but if you have multiple diseases, you may have multiple indications simultaneously, but also the R&D efficiencies that can be realized with this type of approach, because instead of doing a clinical trial for each and every different disease, we basket them together, which can be a great value driver.
Well, it is a delight to have Michelle here in the studio, the CEO of Alterna, the pioneer of engineered tRNA, which I think we'll be hearing more about over the course of the week at JPM and certainly in the years ahead. Thank you for joining us.
Thanks for having me, Christine.